Warm welcome to everyone for today's second lecture exchange presentation about agroecology and its potential and role for food systems transformation. We have again, like last week, two experts who share their perspective with us. The topic today is input reduction. And before I hand over the word to Frank Liebisch and Anna Spescha, I will give a brief introduction to get ourselves all on the same page. So first, I give a short input to get ourselves, okay, where are we? What do we discuss today? Then we will have two inputs of 20 minutes each, both from Frank and from Anna. And you have, of course, the possibility to afterwards ask questions on what you have heard, share your perspective on the things you heard, before we then close the public part and we continue to work with the students in a special session. This public lecture series is therefore also part of a lecture for bachelor students at ETH Zurich within the curriculum, but we are very pleased to open the virtual doors to our lecture room for these 5 public lectures on 5 selected principles of agroecology. Agroecology, with its holistic understanding, has, of course, strong connections to the food systems, which again is strongly connected to the SDGs. If we talk about sustainable food systems, for us, that means food systems need to provide food and nutrition security, address issues of human and environmental health, and of course also connect and support social well-being and livelihoods. And that's why food systems and SDGs as a global set of goals which should or give a direction to sustainable development are strongly interlinked, and it goes far beyond, for example, SDG 2, zero hunger, but it includes a lot of elements from the environment, but also the economics and, of course, the social aspects of sustainable development and the social aspects of food systems. And agroecology, in its definition as being a scientific discipline, a set of practice, but also a social movement, is a nice connex also between food systems as such, with these different dimensions we have to look at, but also to the SDGs. As a science, agroecology studies how different components of the agroecosystems do interact. As a set of practice, it seeks to support sustainable farming systems that optimize and stabilize yields. And as a social movement, it pursues the multifunctional roles of agricultural and food system, promotes social justice, nurtures identity and culture, and strengthens the economic viability of both rural areas, but also more and more the urban food systems as such. And agroecology approaches to favor the use of natural processes by limiting the use of purchased inputs. And that's also the focus of today. We will focus on the principle input reduction and see how scientific contribution can help also to make a link to that given principles of input reduction. But always we have to look at the, let's say, the social ecological systems, because agri-food systems are coupled over these. We do not, there is always a social dimension also in the way we produce, in the way we process, and in the way we consume food. And our kind of basis are these 13 principles of agroecology. Some principles do more address aspects on the agro ecosystem, like the topic of today, input reduction. The aim is to reduce or eliminate dependency on purchased inputs, but there are also principles which address more food system aspects. And last week we talked about connectivity. That's something where more the food system dimension was in the focus. So agroecology does address different levels or different kind of spaces, more farm, landscape or water catchments, but also community and food systems, all the way to societal elements. And it can also be used as an understanding. Do we talk more about incremental aspects changes? That means the increases in efficiency or the reduction of inputs used. But it goes also further up on different levels where questions of redesigning agroecosystem within a landscape perspective are addressed. That goes all up to building new global food systems based on participation, localness, fairness, and justice. So that said, we always have to place ourselves, where are we at the current discussion, knowing that there are different levels, knowing that there are different kind of spaces we look at. And we will talk about other principles in the weeks to come. We will talk next week about recycling, then fairness, and close this series with a view on soil health. But for now, we talk about input reduction, and it's a great pleasure to welcome Frank Liebisch and Anna Spescia to our virtual classroom tonight. Frank is Group Leader Water Protection and Substance Flows at Agroecology, the governmental research institution on agricultural and food systems. And Anna Spescia is a researcher at the Plant Pathology Group here at ETH Zurich. Thank you, Frank. Thank you, Anna, for being with us. And without any further ado, Frank, I am happily stepping back and ask you to join the virtual floor. Thank you very much for being with us, both of you.
Thanks a lot, Martijn. So can you see my presentation?
Yes, nice and clear.
Okay, then I will start and hope I manage my 20 minutes. Yeah, to introduce myself a bit more. Martijn said it. I'm head of the group of water protection and substance flows at Agroecology, and we work on nutrient management and on different scales, being from the process to let's say the national scale. And within this group, we look a lot on investigating consequences of false or erroneous nutrient management, or also on the tools that can help us to do better decisions in the future. On myself, I'm a trained gardener from the very beginning, then became an agricultural engineer studying in Hanover, and came to Switzerland in 2007. There I did a PhD at ETH, and in the group of plant nutrition at the beginning, and until 2019 I was researcher and lecturer at the ETH, and from then onwards started my work as a head of the group of substance flows and water protection. So to start with, I will follow up the introduction to the SDGs. You have seen this wheel before. I just want to remember you what's about nutrient flows, and you see it's about the production, reducing hunger. We have our resources, clean water and sanitation. It's about responsible consumption and production. Climate effects play a strong role, and last but not least, it's a lot about biodiversity and life on land and water. You have seen the levels of transition towards a more sustainable food system. In my talk, I will focus mainly on recycling and input reduction being the two of them, and our work is mainly focusing from level 1 to level 3, I would say. So to stay in time, let's jump into the topic, and I try to arrange my talk a bit on the questions I was given to, and my talk today will not focus on the global scale, but rather on Switzerland, as the title said it in the beginning. But to set the stage, I would say, I want to ask the question, what makes the nitrogen input reduction that important? And maybe maybe most of you know this kind of figure from Steffens and colleagues, where it's clearly shown that nitrogen and phosphorus cross our planetary boundaries quite a lot. And adding on that, we can say that the major perturbation is made by agricultural management. Having said that, I also want to show another slide showing that greenhouse gas emissions from nitrogen fertilizers could be reduced by up to 1/5 with combined interventions focusing on a lot of different loss pathways. And on the very left, you can see basically in black, that's the current global greenhouse gas emissions, which are projected to still increase quite a bit till 2050. And in the middle graph, you can see basically our reduction potentials we can have on different ways and relations to agriculture. And fertilizer substitution is still um, predicted to increase a bit, but with a lot of other ways, we can still reduce a lot. And on the very right, in red, again, you see, so to say, the baseline emission. And by using many different options and combine our possibilities to reduce losses, we can actually reduce this quite tremendously. And that's actually good news. And with that, I want to jump over to Switzerland. So I will focus today on the national scale, giving here and there a few comments on the management scale. So also in Switzerland, a lot of our nitrogen use is linked to resource and environmental problems. And then many of them are strongly related to agriculture. On the left-hand side, you see basically the nitrogen deposition from the atmosphere, which was modeled for Switzerland. And you can see that in the regions being in pre-alpine regions where mostly grassland and animal-based agricultural systems occur, there we find more deposition. That means basically in animal-based systems, we have more ammonia losses to the atmosphere, which later on return when it's raining, for example. And on the right-hand side, you see basically the points which were measured in our drinking water reserves over Switzerland. And you can see that most drinking water problems by nitrate pollution occur in the very intensive agricultural landscape, being slightly above our animal production systems, and causing 15 to 20% of the reserves being above our quality thresholds for drinking water, making measures necessary by law. So to fetch you a bit and then shortly say something about our nitrogen use and flows in agriculture, I want to introduce you to this untied tube model, which was developed or established by ENEMA and colleagues in the EU nitrogen report. And here agriculture is shown as a tube system, we can say. And on the left-hand side, you see the crop production system, crop production systems where you have different types of crops, different varieties, different management options. You have input by fertilizers from the mentioned deposition, biological nitrogen fixation, and so on. And the small arrows you see in red here are a lot of different losses we can have. So we have nitrous oxide, ammonia, ammoniac, um, N2, which is not very dangerous, but also nitrate and ammonium. And on the other side, you have the animal production system, of course, different animals, animal numbers, different management. You can see then here the output of our animal systems being milk, meat, eggs, and so on. And also here, we have a lot of different possibilities for losses. And the crop outputs I did not mention before. Of course, our harvest crop is here. And to the animal side, we have the inputs of yeah, external feed and so on, and our losses can go to the atmosphere and the groundwater. So roughly that you are aware, there are many loss pathways around, and of course, different production systems are interconnected, and we have a lot of different sources where our nitrogen can come from and being lost too. On the next slide, I show it a bit more simplified here as there's the nitrogen balance for Swiss agriculture. On the top left, you roughly see the imported feed being 51,000 tons of nitrogen into Switzerland. We have the animal source food that is produced on the right top side. On the lower level to the right, we have all the inputs, so mineral fertilizers being 46,000 tons, and so on, biological nitrogen fixation and deposition. And we have the plant-based food and products being much less. And in red here below, you see basically the surplus, which is 97,000 tons of nitrogen every year. In Switzerland, indicated here, you can see that the nitrogen use efficiency is quite low on that level, showing that we have basically 93 kg of nitrogen per hectare agricultural area is lost or on the surface. Just to mention it, there are also internal flows. Here you see the manures and slurries, so organic fertilizers, which do not count as input or output per se, because they are produced on the farm, and vice versa. We have also the feed of the farm produced, and you can see that the numbers in there are much larger than the inputs or outputs we find. And that's important when we look later on our efficiency of the systems. So knowing that, I would ask the question: how can we reduce now this nitrogen surplus on national scale, which is roughly as large as our inputs? With this slide, I want to show you roughly the farm balancing system. So in Switzerland, the farmers are obliged to fill a farm-based balance, where they roughly show that the nutrients they have in their farm system are kind of balanced. And there are 4 modules to say it very simple. Module 1 is the feed balance. So what kind of food they produce themselves and the feed they import. We have module 2. This is based on the animal numbers, the manures and slurry they produce themselves. We have module 3, which basically calculates the actual crop and plant nutrient requirements. And module 4 deals with importing or exporting of the fertilizers or nutrients they have. Just to say it, the little symbols with the different colors, they just refer to different databases and sources of knowledge, which can be also self-declaration by the farmer, but also being official databases by the Ministry of Agriculture. And for the first of the 2 examples I will describe here today, we will look into module 2. So basically explaining the nutrient availability within the farm. And this nutrient availability estimates basically the total nutrient availability minus potential losses. And what that means shows this kind of figure where you see on the left hand the example of one large animal unit, which is often still used in policy to describe nutrient flows, which we can use for this example as 100%. And if you take away all the losses, which potentially depending on farm system, farm management, and so on, we arrive on the right side just for the 21 to 56 kg of available N, which basically says that large parts of the total nitrogen is actually accepted being loss. And knowing that, we can say that this kind of balancing system does not give incentives to increase nutrient or nitrogen use efficiency. Having said that, this balancing system is basically 30 years old, and since then in use with just frequent adjustments, being mainly on numbers and on very marginal things. Of course, back in the time, it was a very innovative system, but it hasn't adjusted to changing knowledge so far. And it still uses average values and is based on a very strong simplification of the agriculture or farm system. Having said that, there is a strong need for revision of the system. So what could we do to improve such a balancing system? You see here a feed to crop production cascade, as we say, which can be used to describe the nutrient flows in our agricultural system. And you see on the top left, basically the start with the feeding, bringing nutrients into animal production systems, and these animals then excrete nutrients, and then on the farm you can have the animals in the yard, in the stable, or on the pasture, and your manures and slurries can then go to storage, and right after that they will be spread as fertilizers again, and be in the agricultural field as fertilizers. And still there it does not end, because organic fertilizers have also a release over years, meaning that we can have nutrient flows also in subsequent years, which should be considered. Having this kind of improvements in mind, you see now here an example study where on the very left side you see how the nutrients from organic fertilizers are considered in the actual balancing system of 222 farms. And just by adjusting, so to say, this system from average values towards a cascade model, as I have shown you, would be the step to the second box plot I show you, and would already increase the consideration of available crop nutrient. And if we then go a step further, saying that we could even improve by adjusting it for farm specific numbers and considerations, because each farm has different options to decrease their losses and improve nitrogen efficiency. You would see again an increase reflecting the average Swiss farming model, showing that there is already quite some measures on the way. And on the very right, you see a maximum technical potential scenario showing what would be possible if you use best practice, the most loss reduction measures we are aware at the moment. And knowing that, you see that there is quite some potential that we could reduce the surplus nitrogen in Swiss agriculture management, just by considering the nutrient availability in our organic manures much better. And from that example, I will come to the second example, which rather goes to the fertilizer requirement side on the crop side. And there, this Swiss balance today considers just average nutrient demand and not the actual demand of our crops. What does that mean? So the definition of good nutrient management practice and fertilization or plant nutrition says that we should ensure nutrient supply to plants, taking into account the supply from the soil, also create optimal nutrient ratios, being N and P, for example. We should replenish the nutrients we take off by the harvest from the field. We should align our application rate, of course, to economic optimum, but also to the effects of the environment, basically saying an ecologic optimum. And then we should also improving soil properties and maintain the yield capacity, which basically says maintain soil health. Saying that, it's basically necessary to have a site and situation specific management, meaning average values are not sufficient. And here I just show you a principle how such site-specific fertilizer recommendation could look like. That's from the Swiss Principles of Fertilization. This is this book you see in the lower right side. And basically, you from this average fertilization norm, you go with different factors, considering your yield potential, your mineralization from the soil, your soil management, tillage, your pre-crop, the release from previously applied organic fertilizers, and so on. And then with that, you calculate basically the actual fertilizer demand. Here an example, it's also several hundreds of farms investigated, are shown the main crops of Switzerland being winter wheat, sugar beets, silage and grain maize, and of course some grasslands. And you see again a fertilizer balance, and in green, this would be a surplus on field level now. So you can see that we have quite significant surpluses found in such agricultural systems, and this shows very likely that it is not fertilized according to a site and location specific needs. If you look on these large arrow bars, you see also that there must be quite some farms which work already according to sustainable standards, I would say, on the other hand. But on average, we have still quite some large surpluses in the system. So a more actual example would focus a bit on these correction factors. Here you see several farms from a current project where you now see in colors basically these factors we could correct according to the Swiss fertilizer recommendation. And for 2 years shown on this slide, you can see that in absolutely most cases, we have the theoretical potential to reduce the actual amount of fertilizer being brought to the fields. But looking a bit more specific now on the farm situation, as you know, theoretical potential is not absolute potential. You can see that in some farms we can actually produce or reduce the fertilizer amount and by real numbers, but on other farms, such a correction model would show that we still would need more nutrients. And the reason for that is that we um, did not know for this study the actual yield potential laying behind, but also that the production potential is one of the, let's say, weak options that can change from year to year, and each farm has a different resource and use availability and also soil health. So having so to say a positive nutrient demand in such a calculation could also mean that they have high production but lose soil fertility. And this basically brings us back to the principles of agroecology, where we have to look not only on production but also on long-term sustainability of our resource use. And with that, I will come closer to my final slide and ask, how can we improve now the end use efficiency on national scale? And the two examples I showed basically say that by a more realistic consideration of our organic fertilizer resources, being manures and slurries, could help a lot. And site-specific fertilization would be on the other side of our farm balancing system, can also contribute significantly to a reduction of nitrogen use and subsequent loss in our systems. You have seen this figure, and now you know where we are. So on the lower left and also in the manure side, these are the two of the big levers we have. There we can become much better. Of course, you also see it that feeding plays an important role. This must be another talk probably at some point in time. I could not put it into my time slot, and then I would have need to make use of the expertise of some colleagues of mine. And with that, I come to my almost last slide. So what are the challenges that we face in supporting input reduction or gaining a wider adoption of agroecology? And just the second question as well, what's the role science can play or policy and practice overcoming these changes? And there I would just give the three statements here for the subsequent discussion. I would state that the tools and knowledge for such a reduction is available. We also see in our experiments that practice can do it, but has little incentive to actually do it. And I would say it's about society and policy to change the rule actually, the rules actually, which are behind our legal enforcement of nutrient management. And with that, I thank you for listening, and then all my colleagues that contributed to the data and figures I showed you.
Thank
So,
you very much, Frank, for this input on the different flows. And I would like to ask you to directly unshare your screen so that Anna can start her presentation. And I would invite the audience to keep their questions or write some questions into the chat. And we will open for questions on both presentations after Anna's contribution. Anna, the floor is yours. Thank you for being with us. You're muted, Anna.
Yes. So can you hear me now and see my presentation?
Both very clear. Thank you.
Then thanks for inviting me, and welcome to my presentation about protecting plants from pathogens without pesticide inputs. So I'm Anna Spesha. I'm a researcher at the plant pathology group at ETH, and I recently finished my PhD on biological control of insect pests. And this is also the main example that I will give you in my talk. But first of all, so since 10,000 years, we're breeding crops and selecting them in a way that they're the most tasty for us. But they're not only tasty to us, they're also tasty to a wide range of plant pests and diseases. For example, insects, bacteria, viruses, fungi, and mice also really like our crops and to feed on them. And like this, we lose 70 to 30% of all our yields every year due to plant diseases. And in conventional agriculture, these diseases are mainly fought by spraying pesticides, which is then again linked to a lot of environmental problems. So I think the most well-known example is the use of neonicotinoids. They are super effective in fighting target pests, but they also have a lot of effects on non-target pests, for example, on bees. So I guess you saw some news articles about bee dying due to neonicotinoids, and this is why this pesticide was banned in a lot of areas. And of course, the pests are also happy about this, so we need other measures to control them. And for this, like, there are a lot of alternatives to pesticides, and I don't have the time to go into detail or talk about even more than just biocontrol, but I just want to quickly mention two other alternatives as well, because I think they are also really important. And one is breeding plants that are resistant to diseases. So you select basically crops that are resistant to the pest, and you can also speed up this breeding process by using molecular markers, so that you already know a bit sooner before you have to test it in the field if the plants are resistant. And one institution that is also doing this, but also a lot of other things, is the International Center for Tropical Agriculture, the CIAT. And another really great system are push-pull systems that have been developed as well at ICP, that is also doing a lot of great research in this direction, but also in biocontrol. And the most well-known system is with maize, and you basically plant crops that are repelling the pest with maize, so that they don't really smell the tasty maize, but only this repellent crop. And then you plant a crop around, or a plant around the maize field that is actually really tasty for the pest, so the pest only is in the border and not in the maize field. And what I will talk mainly about is biological control, which is defined as the use of living organisms to control plant diseases. And there are different ways of biocontrol. And in classical biocontrol, you basically introduce the natural enemy of an introduced invasive disease, which can work really well if you have a parasitoid that is very specific, and then it only targets the caterpillar that you have already imported. Then this works really well, but this can also go wrong. For example, we introduced Asian lady beetles here to fight or to feed on insect pests. And the problem now is that they take over the area here, and then our native lady beetles suffer from that. Then there is conservation biocontrol, where you basically want to modify the agroecosystem in a way that you enhance natural enemy populations. One very simple example is the flower stripes at the border of the field, where you enhance biodiversity, and you also have a more diverse insect population, and some of these insects can then feed on the pest in the field. And the third way is augmentation biocontrol, where you release a high number of natural enemies to achieve a temporary disease control. And this is the kind of biocontrol that the research in my group is doing, where we go to the field and bring out a lot of bacteria, and they then can establish in the soil or on the roots throughout the season. But when winter comes, also the bacteria number declines, and then in the next year, the farmer has to again apply the bacteria for biocontrol. But of course, on a farm scale, this is also sprayed in a way you would also spray normal pesticides. So this is just a research image. And the way that biocontrol can contribute to agroecology and also to input reduction, but also I think it also contributes to soil health and biodiversity. So the goal is to use natural resources to control plant diseases. You can have an input, which are the biocontrol agents. So depending on which way of biocontrol you use, you still have inputs, but the main goal is to reduce chemical pesticide use, so that you don't need to spray pesticides that are very harmful to the environment, but to use biocontrol. And like this, we want to reduce negative effects on the environment, because, as I mentioned before, pesticides can have very, very bad effects. The research in my group focuses mainly on Pseudomonas bacteria from the groups Protegans and Chlororhynchus. What is remarkable about them is that they are very competitive root colonizers. I think you can see here a few bacteria cells on a root. And so they are colonizing these roots at very, very high densities. And they are competing against other bacteria and fungi that also would like to live on the roots. They produce a lot of antifungal and insecticidal metabolites, and in this way increase plant health. So there's this nice diagram here where you can see all the positive effects they can have. They compete against other bacteria and antagonize fungi. When insects feed on plant roots, they can then colonize the insect and kill it in this way. They promote plant growth mainly by making nutrients more available to the plant. And they can also induce systemic resistance, which means that the plant is better protected against foliar diseases. I'll show you now a few examples that have accumulated over the last few decades in this group. So to the left, you see that the Pseudomonas inoculated plant is growing larger than the ones without the bacteria. And they can also suppress pathogens. So to the left, there is a tomato plant infected by Fusarium. No, on the left, there's a healthy tomato plant. And in the middle, there's the one that is infected by a pathogen. So it's heavily reduced in growth. And if you then add the Pseudomonas bacteria, then you see that the plant is growing normally, more or less. Still a bit reduced, but then again, it suppressed the pathogen very effectively. And this holds also true in a lot of different systems, as we saw in this group over in a lot of laboratory and greenhouse experiments. And around a decade ago, it was like first proven that they also have oral insecticidal activity against these 3 insect pests. So they were feeding larvae with bacteria. And we can see here the survival. And we focus here on this CHA0 strain that has an insecticidal activity. So after 1 week, only 0 to 20% of the larvae are alive. And these are very, very nice laboratory assays, and you sometimes also see it in the greenhouse. But the field application of this Pseudomonas bacteria is still very limited, even though there is a lot of research going on with this bacteria. And I think it's a very good example to focus on the challenges that are faced in biocontrol. So one is the efficacy, because often in the greenhouse, an agent is very, very efficient in controlling a disease, but in the field, then the environmental conditions also play a role, and then sometimes it's not very efficient or not efficient enough to give a good protection for the farmer. And another problem is the stability and reliability, which is also linked a lot to the environmental conditions, because maybe in 1 year, the Pseudomonas bacteria have like perfect conditions and they can really suppress the disease a lot. But then in another year, it might be too dry or too hot, and they do not work. And this is really important, that when a farmer uses biocontrol agents, that it really works reliably and not fails half of the time. And because we speak about living organisms, it's very important, but also very difficult to find a product formulation with especially a long shelf life, because of course, when the bacteria are just standing around on the shelf for a long time, they also die or are less effective when they're brought to the field. And the last major challenge that I see is product registration. At the moment, it's like the same process for pesticides as for biocontrol agents, which is a problem because biocontrol agents, they're living organisms, they function differently to pesticides. So they should also have like a different regulatory process. And this is something that really needs to be changed, hopefully soon, so that there is like a clear requirements, which safety assessments need to be performed when you register biocontrol agents, and that it's not like the same as for a pesticide, because it's very big difference if you spray a pesticide or whether you apply a biocontrol agent that produces toxins as well, but it produces it very locally on the root, or even some toxins are only expressed inside the insect. So yeah, this really needs to be treated differently. And in my project, I focused on the first 2 points on the efficacy and reliability that we wanted to improve by combining different biocontrol agents. And in order to present you my project, I also have to present you all the agents that I was working with. So the Pseudomonas I already introduced. And then I was also working with entomopathogenic nematodes. Entomopathogenic just means that they're killing insects. And the nematodes are living in the soil and actively hunting for insects. Once they find them, they enter them and release their associated bacteria that then kill the insect. The spores of entomopathogenic fungi are laying around in the soil, and when an insect passes by, it attaches, like the spores attach to the skin of the insect, and then breach through the skin and kill it in this way. These three different biocontrol agents have very different modes of action. So they enter the insect differently, but they also produce different metabolites and toxins. We think this could lead to combinations being more efficient or more stable. So when the insect is attacked by 3 pathogens at the same time, it of course it's much more difficult for it to defend itself against these 3 pathogens compared to if only 1 pathogen attacks at the same time. And we also think that it might be more stable. For example, if the Pseudomonas have a really hard time in the field in 1 year, the nematodes or the fungi might still have good conditions for them and might work. So kind of if one fails, the others take over. So my first research aim was to develop a biocontrol consortium. And the first task focused mainly on the Pseudomonas bacteria because they were so far not applied against root-feeding insect pests. So we first wanted to test whether this is possible, and then combine them with nematodes and fungi to increase efficacy and stability. We took a very stepwise approach with first doing laboratory and greenhouse assays, then doing pot assays outside, and in the end, a field trial. And I will now present you the results of my field trial that I did with the cabbage maggot Delia radicum. It's a very important pest in brassicatian crops, for example, for cauliflower or rapeseed, but also radish. And you can see here the damages that this insect produces. So there are this very specific mining that it leaves. And we had this field where we sprayed our biocontrol agents, and we of course also had a control without application. And we then rated the damage. And the green part is where the plants were not damaged and which can be sold for a good price by the farmer. So in the control, around 40% of the plants are not damaged by Delia radicum. And all the biocontrol agents increased this amount of non-damaged plants so they could protect radishes. And especially the Pseudomonas and the triple consortium were very effective and led to an increase by 50% compared to the control. So to sum it up, we found Pseudomonas, nematodes, and fungi that are effective in controlling the cabbage maggot. And when we looked at the soil and root colonization, we saw that they are also compatible with each other. When we compared the consortium to the single agents, we saw a few synergistic effects in laboratory and semi-field assays, but in the field, the consortium was only as good as the Pseudomonas alone. And to give a brief outlook, to really determine if this consortium is more reliable than the single agents, we have to, of course, do a more field experiment to really be able to say this. The second aim of my thesis was to have a better insight in how the Pseudomonas, nematodes, and fungi interact when they infect the same insect. So we did laboratory assays with 2 insect pests, with the large cabbage white that is feeding on the leaves of cabbage plants, and with the banded cucumber beetle that is feeding on the roots, especially of maize plants. And when we looked at the survival of the larvae, we saw that the triple combination is always the fastest in killing the larvae. And compared also to the single agents, it was just much faster in killing them. And we also looked what was going on inside the larvae, and we saw that they can co-infect, and also in the first stage of the infection that all the biocontrol agents are present and playing a role in killing the insect. But the further the cadaver decayed, the more the organisms competed for resources, and in the end, the Pseudomonas dominated the infection. So we hope that we have discovered a consortium that has a very big potential to be applied against a variety of root-feeding insect pests. And we would like to see that when we apply that, thanks to the Pseudomonas, they can also control soil-borne fungal diseases. However, we need a lot of further research. So we did one field trial against one insect pest. So we need to do much more field studies in order to really be able to say this. And of course, once we have completed this, we would need to assess the safety of our agents and then develop a product that then could be registered for biocontrol. And of course, for the product development, we would also need to decide whether we want one product with 3 agents or like 3 different projects that we just recommend for applying in a certain, like together or after each other. And with this, I'd like to thank you for your attention. I'm really looking forward to the discussion session.
Thank you very much, Anna and Frank, for sharing some of your insights. And it's now time to open the floor for questions to either Frank or Anna. Please raise your hand, or yes, Nina, please.
So that was very interesting. Thanks a lot. I actually wanted to clap and not necessarily raise my hand, but there are always lots of questions. So Anna, thanks a lot with these field trials. How easy is it actually to do that outside? I mean, I can imagine that these field trials, or that the trials are easy to do under controlled conditions, where you also can control who's flying into whatever your greenhouse or your climate chamber. But how much of the interactions do you get when you do such experiments outside?
Yeah, so I did one field trial and we just had like the perfect conditions for this one field trial and everything worked perfect. We had a lot of pest insects that were flying in and laying their eggs in our field. But of course, it's very important, and not every year is such a great year for the pest. And also, it's of course a lot of effort to rear the biocontrol agents in the lab and time everything. Mm-hmm. But it would be a lot of effort to conduct field trials at multiple sites in order to also, control for the different environmental conditions, but also pest incidences. Mm-hmm.
Wow.
And it really depends how high the pest pressure is in a field trial. Yeah.
Right. Okay. Thanks a lot.
Thank you. Thanks. Other questions to Anna or Frank? Yes. Dieter Müller write something in the chat. I can translate, but you're also free to directly ask the question. So what are the thoughts about the field trial and lack of success with combined biocontrol?
Sure, there must be some hypothesis. What went wrong with compared to the greenhouse trial?
So actually, I think for me, the combination worked really well in the field, and I'm not super surprised that I didn't see like a higher efficacy of the triple combination, just because the Pseudomonas treatment was very, very effective, which I've also seen in other assays. So I think it really depends a lot on the conditions, whether we are able to see synergistic effects or not. And but this is like just one project, and in general, I think a big problem of using combinations in biocontrol is that it is very labor intense to really test the consortium that is then not inhibiting each other, which I think, most of my time was spent on doing this evaluation. And then also to formulate it, and then it's also more expensive to buy 3 products instead of just one. Yeah, I hope this clarifies it.
Thank you.
Jana, and then there is a question in the chat from Cyril, but first Jana, please.
Yes, I have a question for Frank. So you mentioned before that the balancing system has been in use for quite a while, but it hasn't really been adjusted yet, even though that's a very necessary step. Could you tell us why? Is it more, I don't know, a lack of motivation, or is there any other factors playing into this?
Yes, I can also speculate a bit. So I'm changing a balancing system which is valid for a whole nation, and then for 50,000 farms is always an effort. And then you have strongly to balance your decision. Do you want to change something where people have to get adjusted to, which has to be accepted, and so on. So the Swiss balance in principle was very innovative in the beginning, in the 90s. And of course it was adjusted a bit, but it was mainly a bit of numbers and here some correction of animal excretion and so on. So rather small things. But after 30 years, you can imagine that our knowledge is much better. And when it was introduced, it was basically a tool of simplicity. So the idea was that every farmer with his calculator and the sheet of paper could calculate it. But today we are able to calculate the Swiss balances for all 50,000 farms with our own mobile phone in terms of calculation power. So also this kind of things have changed, but the basis to let's say digitalize this is a stronger decision. So we can only speculate. It's a mixture of lack of motivation, but also not to change too fast. Take all of the farmers with us, and so on. So there might be a lot of different reasons, including also politics. So in Switzerland, decision on agricultural politics is heavily debated every 4 years at least. And but now is some time, I would say. So the digitalization is strongly underway, and then a lot of things also in our knowledge change. Does this answer your question a bit?
Yes, it does. Thank you very much.
Pleasure.
Thank you, Jana and Frank. There was, as I said, a question in the chat from Cyril, whose microphone is not working. So I do quick the translation. So first, thank you for the talks of both of you. And there is a question for you, Anna. Are there products on the market and how competitive are these also in an economic terms compared to pesticides?
So yes, there are products on the market, especially for the fungi and nematodes. There are a lot of products on the market. For Pseudomonads, there is much less. There is only a few products, and in Europe, they're not registered for insect biocontrol, but only against pathogens. And also, they're not registered for combined use. And I don't know how much it is actually recommended by, for example, companies that sell them. Then for how competitive they are economically, I actually don't really have good numbers. So I think they're not super competitive yet. But yeah, I think it will become more competitive because like the production, large scale production and product formulation is increasing or getting better like all the time. So, and also with more and more pesticides being banned, there is just at some point you just have to have an alternative because you cannot use pesticides anymore. So I think, yeah, there are two main points here.
Oh, sorry, other questions to Anna or Frank? So Cyril seems to be very happy with your answer. That's good news. If there are no questions, I would have one, Frank, to you. Maybe I didn't get it right, but if you explained a view, a perspective, the Swiss balance, 50,000 Swiss farms. You said that there is probably a potential over kind of a surplus in nitrogen, mostly on farms with animals. But if you would take single farms and not an average of everything, how accurate is then the assessment? Is that because we have just accumulation of single measurements and that getting them very inaccurate because the diversity of the farms are big, or is there already a huge error on the farm assessment?
So the national scale balance I showed is based on statistics. So these numbers should be realistic. We just cannot say how these numbers are distributed on single farms. But if you go on single farms, it's never that easy. Huh? So this is, I think you understood it right. So it depends on the farm structure, if your major is cropping or if you have a focus on animals. It depends on the area you have where you can spread your fertilizer. So on farm and field level, you have let's put it this way: our national scale balance says the numbers, and then they should be realistic. But this doesn't help us to enforce sustainable management. The sustainable management you have to decide on farm and field level, and that will be farm and field specific. So the decisions which we need should be also based on information you have on that farm. So there the variation can be much larger, but still, so we know that farms with little area where they can spread fertilizer, but large numbers of animals will have a bigger problem working sustainable than vice versa. So the most important thing is that we should focus our management, but also legal enforcement for nutrient management on farm and field level based on our good agricultural practical knowledge. So getting better, that's the main message.
Thank you for this final message in that sense. Are there any other, one last question to Frank or Anna? This does not seem to be the case. So if we would be in an audience room, you would hear a big round of applause, of course. Thank you very much for being with us. Now you have a few icons at least. That's also a nice kind of gift. Thank you very much for being with us. And now I would invite the non-student participants to leave and invite the students to stay here for more kind of closed question and answer round with the two speakers. Thank you very much and see you next week on Monday again. Thank you.